So, welcome everyone to the April speaker series for the Institute for Precision Medicine. We are very excited today to have, when we think about it, one of the leaders in precision cancer medicine. Today we have Todd Golub, who's director of the Broad Institute of MIT and Harvard and a founding core member of the Institute. Todd received his BA from Carleton College and his MD from University of Chicago Pritzker School of Medicine. He completed his internship, residency, and fellowship training at Boston Children's Hospital and the Dana-Farber Cancer Institute. He joined the faculty of Dana-Farber and Harvard Medical School in 1997. At that same time, he served as the leader of the Cancer Genomics in the Whitehead Institute, MI Center for Genome Research. Todd is a well-known, I think, as he needs very little introduction, is a world-renowned physician scientist. I looked up his citations just before getting there. He has over 250,000 citations of his work, pretty incredible. And he's really pioneered the application of genomics to cancer biology and therapeutic discovery. One of the first researchers to use genomic approaches to molecularly characterize human tumors, laying the foundation for what we now know as the use of genomics to classify, diagnose, and treat cancer. And throughout his career, Todd has advocated for the free, rapid sharing of large-scale genomic data and tools to accelerate the pace of biomedicine, and is deeply committed to patient-focused research. And I think one of the things we really do appreciate is what the Broad has done in terms of, DepMap and all of these projects that have generated these incredibly valuable free resources that have undoubtedly accelerated cancer research. If you have any questions, this is webinar style, so put them in the chat and I will be asking them at the end of the seminar. And in his talk titled Perspectives on Cancer Precision Medicine, Todd's going to talk about new genomic tools and approaches that have a lasting impact on cancer research and drug discovery. Todd, thanks very much for taking the time to do this.
Great. Thanks so much, Adrian. It's great to be here. And I have to say, of that introduction that you just gave, I think my favorite part was your calling out our commitment to sharing data, because that's, perhaps the single thing that I'm most proud of. And, at the beginning of the field of cancer genomics, it was not the standard to do that. That is to make all of the underlying data available at the time of publication or before, and it now is. And I'm proud of having been part of that push of our own community and of journals to require it. So thank you for calling that out. So great to be with you, even if virtually today. Just a few disclosures. I'm not going to talk about any of these companies as part of my talk today, but I do have these sources of funding and extracurricular activities. So this, to an Institute for Precision Medicine, this concept of cancer precision medicine needs no introduction. That is the idea that a tumor would be characterized molecularly, using the word genome kind of loosely here, not just to mean the DNA sequence, the somatic genome, but the molecular attributes of a tumor that would somehow be used to predict the right treatment for a particular patient at the right time, that would lead not just to some, transient, non-meaningful response, but for a durable clinical response, if not cure. That's the holy grail of the cancer precision medicine concept. But I think if we hold a mirror up to, where we are in this field in 2023, I think it's fair to say that still most patients don't really benefit from this paradigm, at least in its fullest form. And so, a reasonable question would be, well, maybe the concept's wrong, and that we should be pursuing an alternative concept of, treatments for all patients, regardless of their particular molecular subsets and so on. I believe pretty strongly that this is the path to the future for all patients, and that it would be a huge mistake to give up on this paradigm, and still recognizing that there's a lot of work to be done because most patients don't benefit from it. So I'm going to sort of give some musings about different approaches that we're taking to pursue new avenues into cancer precision medicine, that one could loosely group into two basic buckets. learning all you can from patients and learning all you can from laboratory experiments. And I think, just cut to the chase here, I think the right answer is that it's not a matter of which of these is better or more powerful. It's how do we use them both and integrate the data together. Learning from patients, obviously, it's the most physiologically relevant model system one could think of, the patient themselves. And yet there are certain things that you can't do in patients, like do perturbation studies of, treat with all drugs in a given patient to see what happens. And so they're limited in that dimension. Those kinds of systematic perturbations or drug treatments or genetic perturbations, of course, you can do in the laboratory, but then you're limited with the very imperfect nature of tractable cancer models like cell lines. And so those are imperfect as well. And so I think the key for the long run is going to be to figure out how do we generate both types of data and learn what we can from each, rather than relying on the perfect method or the perfect model. So let me start on the clinical side, because this is an area that I continue to think is potentially going to be quite important, both in oncology and beyond oncology. And that is really learning all we can from the patient experience. As this audience, I'm sure, knows, in the world of oncology, the field is challenged by the fact that, most patients aren't treated at the Pitt Cancer Center or Sloan Kettering or Dana-Farber or MD Anderson. They're treated out in the community, and most are not treated on clinical trials. And so as a result, most cancer patients don't have the opportunity to participate in research. And yet, if you ask them, how do you feel about that? They say, actually, we're frustrated by that. No one asks us, whether if they're seen by a community oncologist, no one's asked us whether we would like to be participants in a research study. And that's for good reason, because busy clinicians, there's not really a mechanism to incorporate research into that clinical practice. And so the question that a number of colleagues at the Broad Institute and Dana-Farber in particular started asking was, could we use emerging technologies, and in particular social media, to engage patients directly, not through their institutions, not through their care providers, but directly to engage them as partners in research, and doing that, of course, with the highest standard of informed consent, which is now possible to do entirely online with good understanding. And would it be possible to reach patients outside of the major academic cancer research centers? With a goal of this initiative, which we call Count Me In, being to make it possible for any patient anywhere to raise their hand and contribute their genomic and clinical information to research. It should be as easy as that. Declaring your interests, obtaining informed consent, and pressing a button that makes it possible to share your data if you want to. And this work was led by Nick Wagley at the Broad and Dana-Farber, who's recently left to take a leadership position at Genentech. And just want to show some of the earlier output of this Count Me In project, starting with metastatic breast cancer, which of course is a big clinical problem. And yet the number of patient samples, for example, somatic genomes that have been sequenced and made publicly available for patients with metastatic breast cancer, to be distinguished from breast cancer at the time of diagnosis, localized breast cancer, which is a great focus of the TCGA and the International Cancer Genome Consortium. There's not a lot of data publicly available for metastatic breast cancer. And so Nick and his colleagues asked, could we stand up a website, use social media and patient advocacy groups to reach these patients where they are and engage them as partners in research? And within the first year of trying this experiment, there were metastatic breast cancer patients from all 50 states in the country who had enrolled in the study. making their clinical data available, making germline DNA sequence available, and making tissue blocks available via the pathology departments that held them. I think what's most interesting about this isn't so much just the numbers, but where these patients came from. So this is looking at the first 17 or a couple thousand patients that were enrolled in the study. And what was most remarkable here is that while yes, tens of patients came from the biggest cancer centers, MD Anderson, Dana-Farber, and so on, there are over 1,200 institutions, like a clinical practice, that were represented by just one patient. And that, I think, is kind of contrary to the way we typically think about clinical investigation, where you have large studies that are anchored at major centers contributing a lot of patients. Here we have, these single patients coming from across the country. And I think this is something that we're interested in scaling significantly to different cancers, particularly those cancers that are not readily studied at single institutions. Rare cancers, for example, there was an angiosarcoma, count me in, project that it only has about 300 patients per year diagnosed with angiosarcoma. Within 3 months of launching an angiosarcoma project, there were 300 patients enrolled in the study. That led to the discovery of high-frequency mutations that predicted potential response to immune checkpoint blockade. Looking at the clinical data, indeed, there were two patients that were treated with checkpoint blockade off-label with these cancers that responded, and that has led now to a clinical trial studying checkpoint blockade in angiosarcoma. So really a very rapid movement from a rare disease that no institution saw enough of the cases for to a clinical trial testing a particular hypothesis. So I think this is something that I'm excited about scaling, again, for rare cancers and for rare diseases generally, not just cancer, because when you think about it, rare diseases, each rare disease may be rare by definition, but collectively it's estimated that as many as 10% of individuals in the US suffer from one rare disease or another, but no institution sees enough of those patients to be able to really do a major study. But if you could develop patient registries and genomic and other kinds of molecular information and coupled to clinical data in a more distributed, direct-to-patient manner, that could really change the face of how we think about rare disease generally. I want to turn now to the other side of not learning directly from patients, which is, of course, important, but learning from systematic studies in the lab. And Adrian mentioned the cancer dependency map. So I'd like to mention that for those of you that aren't familiar with that. And this is a project led by Paquita Vasquez at a senior scientist at the Broad. And the concept here is to take large numbers of patient-derived models, cancer cell lines, comprehensively molecularly characterize them. So now we're at about 1,000 cancer cell lines that have been sequenced, genomes have been sequenced, RNA sequenced, some extent of mass spectrometric proteomic analysis, metabolite profiling, methylation analysis, and so on. So deeply molecularly characterized, and then subject each of those cancer models to genome-wide CRISPR knockout screens to identify the genetic vulnerabilities or dependencies, and also treat them with small molecules to identify, in particular drugs, to discover their patterns of drug sensitivity so that we get a map of cancer dependencies. And then once you have that matrix of data, systematic matrix of 1,000 cancer cell lines in one dimension, deep genomic characterization in another dimension, and then in the third dimension, the phenotypic characterization in response to knockout of all genes and what, as I'll show you, treatment with most FDA-approved drugs. Then you get this really interesting matrix of data, which we've made publicly available through the depmap.org website hosted at the Broad, whereby you can use simple or more advanced machine learning methods to develop predictors of particular vulnerabilities, whether it's a genetic dependency or a drug sensitivity. And that, of course, is useful because those molecular features that are predictive of a particular cancer dependency can both provide mechanistic insights into why that dependency exists, and they may inform the development of predictive biomarkers that could be used in the future in the clinic to identify relevant patient populations. So I want to give one example in some detail of our work following up just one of those dependencies that we discovered from the DepMap. And this is the work of Daniel Bondison, a postdoc in the lab who's now stayed on as a staff scientist interested in ovarian cancer. And he asked the question, can we look at this DepMap data and look for examples of genes which, when knocked out, are selectively lethal to ovarian cancer cells compared to all other cancer types? Because, of course, a gene which, when knocked out, kills all the cancer models, you could say, oh, maybe that's the miracle universal cure for cancer, but more likely that's just a universally toxic maneuver that should likely be avoided. But here we're looking for what are ovarian selective cancer dependencies, and we're drawn to one of these outliers called XPR1, which I'll say more about, and I'll come back to in a few minutes. The close behind a gene called Kiddens 220, I guess a second gene that I hadn't heard of before. What's XPR1? It turns out that XPR1 is the sole phosphate exporter in human cells, a transmembrane protein responsible for export of inorganic phosphate out of the cell. The next obvious question using these data are, okay, but are there, in addition to being just of ovarian lineage, are there predictive biomarkers of XPR1 dependency? And there, I'm not showing the raw data, but it turns out that it was expression of a gene called SLC34A2, where high levels of expression of this gene were predictive of genetic dependency on XPR1. What's SLC34A2? It turns out it's a phosphate importer, another transmembrane protein that's responsible for import of inorganic phosphate into the cell. So that makes a lot of sense that if you have high levels of phosphate import, you are uniquely dependent on phosphate export for survival. It's also interesting that it turns out that it had been determined by others that SLC34A2 is under the transcriptional regulation of the lineage specific transcription factor PAX8, known to be essential for the lineage-specific survival of ovarian cancers and the fallopian tube cells from which ovarian cancers are derived. We now look in more detail in a panel of cell lines comparing the XPR1 dependency among those ovarian lines that are XPR1 high. You see that they're highly dependent, whereas if they have low expression of the biomarker SLC34A2, remember the importer, they are resistant largely to knockout of XPR1. So this helps to solidify the hypothesis that XPR1 is an ovarian cancer dependency with the further predictive biomarker of the phosphate importer SLC34A2. We went on to do immunohistochemistry to show that, in fact, this phosphate importer is broadly expressed in a significant subset of primary ovarian cancers, as shown on the left, and furthermore, that XPR1 itself is frequently the target of gene amplification in ovarian cancer. So that starts to feel good, that you have selective amplification of the target XPR1 and common high levels of expression of the predictive biomarker SLC34A2 in a substantial number of ovarian cancers. So the model that we've developed as a result of this is to say that in normal cells, you have this equipoise, the balance between phosphate uptake through the SLC transporter and phosphate efflux through XPR1. But in ovarian cancers, as shown on the right, where you have PAX8-driven accumulation of the importer SLC34A2, this must be balanced for the cell to remain in phosphate homeostasis with an accumulation of XPR1. And so if you then were to inhibit XPR1 function genetically or in principle pharmacologically, you would have collapse of the cell through phosphate accumulation, which we've shown biochemically is in fact the case. So that is all internally consistent. It doesn't exactly address the question of, well, okay, but therapeutically, how are you going to accomplish this in patients short of XPR1, in vivo gene editing therapy, which at the moment, there aren't really delivery mechanisms to make that possible. One approach, of course, I'm going to skip that. One approach, of course, is to just make an antibody against XPR1. We're trying to do that. Turns out technically that's quite difficult. But Daniel Bondison in the lab also made this interesting observation that a critical domain of XPR1 called the XPX domain, which is required and necessary for phosphate export activity, is de-repressed by binding of inositol pyrophosphates, in particular, inositol 7 and 8 pyrophosphates, which are themselves synthesized by an enzyme called IP6K, inositol phosphate 6 kinase. So, and that's been shown by others to be the case. So the question was, well, would an inhibitor of IP6K recapitulate the effect of XPR1 genetic loss of function? Could we induce this conformational change in XPR1 that would result in loss of its phosphate export function? And it turns out that there is an IP6K inhibitor that was developed by a Japanese company called Scoia Pharmaceuticals that wasn't thinking about this for ovarian cancer, but rather was trying to develop a drug for hyperphosphatemia. The structure is shown here. We synthesized this compound SC919 and then tested it in a panel of ovarian cancer cell lines, as shown on the right, both those that express high levels of the predictive biomarker SLC34A2, as shown at the top, and those that are negative. And what you can see here is that in each of these cases, if the predictive biomarker, the importer, phosphate importer, was highly expressed, you see a quite dramatic killing of those cells when treated with the IP6K inhibitor, whereas for those cell lines at the bottom, which don't express the biomarker, they don't show sensitivity at all to this IP6K inhibitor. So that's encouraging that, maybe there's a path to think about IP6K inhibitors in the future for translational therapeutic testing in these ovarian cancers, specifically with high SLC34A2 expression. Now, I'm going to come back to this compound in a second, but first I want to come back to a method that we've developed in thinking about the power of the genetic screens that I showed you. So again, the DEP map is 1,000 cancer cell lines in which 20,000 genes have been, actually it's only about 13,000 genes, have been knocked out. You can only do that because of the power of pooling, of a pooled genetic screen. One library that has guide RNAs targeting all 13,000 genes. It'd be great if you could do that with small molecules. There's not really a great way to pool the small molecules themselves, but what if you could use a clever pooling strategy to pool the cell lines themselves? And so Channing Yu, when he was a postdoc in the lab, developed a method that he called PRISM, which is a simple concept. Could you introduce into each cell line in the DEP map a unique molecular barcode? It's a random 26 nucleotide sequence, randomly integrates at about single copy into the genome of these cancer cell lines. Now that they're molecularly barcoded, pool all the cell lines together, and then treat these pools with, for example, a drug of interest, and then measure the abundance of each of the barcodes before and after drug treatment. And you can do that measurement either by sequencing, or in high throughput mode, we're using Luminex beads that are coupled to an anti-barcode tag for efficient high throughput detection of and quantification of these barcodes. And so the idea here is that if you see following drug treatment that one of the barcodes becomes depleted following treatment, you can infer that the cell line that was tagged with that barcode must have been selectively killed by the drug that was used in that treatment. So that's the PRISM concept. So now let's apply that to take this drug, SC919, which I showed you in this limited sort of slice of a few ovarian cancer cell lines, looked promising as a selective IP6K inhibitor, perhaps not with without a lot of off-target effects. But what if we put it into PRISM, where we now are profiling about 800 cancer cell lines with this compound, and then comparing the profile of killing across those 800 cell lines with our compound with the killing profiling of genome-wide CRISPR knockout screens that's part of the DEP map, and ask which of the genes, which when knocked out, best phenocopies the pattern of killing by this drug. And the answer, and so here's the curve. It actually is a curve. It's just a big break because that's how clean the data are. That the number one best correlated knockout with sensitivity to this drug is XPR1 knockout. And the number two is that funny named gene that I mentioned before, Kittens 220, which we actually showed is a protein-protein, forms a protein-protein interaction with XPR1 and is responsible for normal XPR1 localization and phosphate export function. So this is a really, I think, cool result because it tells you a couple of things. Number one, shows you how powerful it is to be able to sling around all of this genomic information, CRISPR knockout data at scale, and small molecule data, in a pretty straightforward way once you have the data, and you can do these kinds of analysis, freely to anybody on the DEP map website. But it also tells you a lot about this drug, which is that it's essentially as selective as XPR1 knockout itself. So I think one can conclude from this that there's not a lot of off-target effect of this particular drug, at least as it relates to cell viability across 800 cancer cell lines. And so that gives you a level of confidence in thinking about bringing, whether it's this compound or drugs of this class, forward to the clinic. This is the kind of molecular profiling that we should be doing, I believe, routinely on any drug that enters into a first-in-human clinical trial. We should have this kind of preclinical, intel on its degree of sensitivity or selectivity or lack thereof before going into the clinic. And I believe that if we did that, we have a better handle on unexpected toxicity in the clinic. So this is using the PRISM method to treat 800 cancer cell lines with a single drug. Well, that's just a single drug. What we'd really like to do is to take a panel of drugs, ultimately all drugs, and also profile them across the entirety of the PRISM collection. And so this was the ambition of Stephen Corsello, who just left to start his own lab at Stanford, physician scientist, oncologist. And he quickly discovered that, one challenge is actually that there doesn't exist a single collection of all FDA approved drugs or drugs in clinical development. And so he set out to assemble and annotate such a collection. We call this now called the drug repurposing hub at the Broad Institute. And he assembled this collection of now close to 8,000 compounds, mostly FDA approved, some in clinical development, some approved by European regulatory agencies. I don't have the data to show here, but we had to reach out to more than 75 chemical vendors to source all of these. You'd think that these would just be readily available from a single source. It's not. More shockingly is when we went to verify the identity and purity of these compounds from reputable chemical vendors, a substantial number, about 28% of the compounds failed that QC and had to be resourced before entering into the collection. And so I think the chemists, on this Zoom, if there are any, would say, yeah. And why were you surprised by that? Of course, you need to validate that a compound is what you think it is. I think most biologists aren't used to thinking that way and don't typically do that routinely, but it's super important, because if 28% of the compounds are degraded or the wrong compound, developing a reference data set like this is going to be deeply problematic. So we fixed all that. And so Stephen, as his postdoc project, did a massive PRISM experiment, which you can do because of this pooling barcoding strategy, where he took about 4,500 of those compounds and put them through initially at a single dose, and then with some follow-up screening, tested all those compounds initially at 2.5 micromolar concentration across 578 cell lines, and asked whether did any of them have activity in this PRISM, crazy PRISM assay. And of course, he rediscovered that chemotherapeutic drugs have broad activity. Okay, that's nice. We knew that. He discovered that targeted cancer agents also showed activity. But what was more surprising and interesting was that there were a number of non-oncology drugs, that is, drugs that were developed for some other clinical indication, that show evidence of killing at least some of these cancer cell lines, at least at this initial dose of 2.5 micromolar. And so those are the ones that are most interesting to follow up. These data are also available now on the DepMap portal for those of you that are interested in looking at them. Obviously, we could only follow up a minuscule number of these stories. I'll show one of them that we're continuing to pursue, but first, just like, what do you see when you take a high-level view of this PRISM data set? Again, it's a matrix of 4,500 drugs by 578 cell lines. And if you do a simple UMAP dimensionality reduction to look at the overall data set, you see that these compounds cluster together by mechanism, not based on their structure, but simply based on their pattern of killing across this panel of 578 cell lines. For example, here at the bottom in orange are all the EGFR inhibitors. The MEK inhibitors are here at the top. Microtubule polymerizers cluster together. What you also see is even very weak signals. You can't tell it's weak here, but for example, glucocorticoid receptor agonists, which have very subtle but distinctive growth inhibitory activity across a subset of the cell lines. you see that mechanistic clustering. So while I think glucocorticoid receptor agonists are not, the next great thing as cancer therapies, as a way to get to mechanism, the DepMap and this PRISM data set is turning out to be quite interesting. I want to show just one example that we're following up, and that is of the multi-drug resistance gene ABCB1, its official gene name. This has been known for decades to be a resistance mechanism for a number of classes of drugs. For example, the taxanes in particular, for example, paclitaxel, where where cancers that are selected to overexpress the MDR1 efflux pump are resistant to those drugs, whereas they might otherwise be sensitive. And we can recover that, in the PRISM data set, where what I'm showing here on the top is MDR1 expression across 578 cell lines, where high levels of expression is shown in red. And then below that are examples of known drugs that show this MDR1 pattern of sensitivity and resistance. That is, if they express MDR1 at a low level, they're sensitive, for example, paclitaxel on the bottom, whereas those cell lines that express high levels of MDR1, shown in red at the top, are resistant, shown in white on this heat map. Okay, that's not discovering anything new, but makes you feel good about the quality of the data set. What was interesting and exciting was the discovery of a single compound called tepoxylin, which had the opposite effect. It only killed those cell lines that express high levels of MDR1, but spared those that are MDR1 low. What's tepoxylin? Here's the structure. It turns out that it's currently FDA approved for use in dogs. It was initially developed as a dual 5-lipoxygenase cyclooxygenase inhibitor for pain relief for arthritic dogs. It initially went into clinical trial in humans, but then was dropped, I think, for strategic reasons. We were able, so the obvious question here is, is this MDR1 related activity explained by the cyclooxygenase and 5-lipoxygenase inhibitory activity? And I don't have time to show the data in any detail, but the answer is clearly no, because other COX inhibitors or lipoxygenase inhibitors did not have this activity either alone or in combination. And so that raised the question, well, then what is the mechanism of action? And is there any actual mechanistic connection to ABCB1, the transporter MDR1, or is that just one of many, many predictive biomarkers that isn't related to mechanism? And so for that, we again turn back to genome-wide CRISPR knockout screens, which take this form, which is to take a cell line that is sensitive to the drug tepoxylin, first introduce a genome-wide CRISPR-Cas9 knockout library, then treat with the drug, in this case tepoxylin, and then look for those cells that emerge from that drug treatment as being either resistant or particularly sensitive. That is, those guide RNAs dropped out. And we did this both in gain of function with CRISPR Cas9 knockout and with CRISPR-A, CRISPR activation, gain of function. So activate the expression of 12 or 13,000 genes in the genome and ask, what do we see there? And this is pretty striking, where we basically got one result. Shown on the left is the result of the CRISPR knockout screens, showing that if you knock out ABCB1 itself, this is MDR1 itself, you completely rescue these cells from treatment with tepoxylin. And moreover, if you take a cell line that is otherwise resistant to tepoxylin and overexpress ABCB1, more so than pretty much any other gene in the genome, you can confer resistance. So at least to a first approximation, these experiments show that MDR1 or ABCB1 is both necessary and sufficient for sensitivity to this class of compounds. So, we're still working up the precise mechanism of action here. Because it's a work in progress, I don't have, polished data slides to show here, but it's looking like the mechanism of this compound may be related to iron transport, and that is that iron-loaded compound may serve as a substrate for the MDR1 efflux pump, leading to intracellular iron depletion and cell death in those cells that are MDR1 high expressing cells. I should also make, in case it wasn't obvious, this shows also that the compound is not functioning as an MDR1 inhibitor, because if you knock out ABCB1, either genetically, as we show here, or pharmacologically with an MDR1 inhibitor, you actually rescue the function of the drug. You don't phenocopy the drug. So it's not simply an MDR1 inhibitor. So I think because I want to make sure we leave plenty of time for questions, maybe I'll wrap up. I've shown a couple of vignettes to try to illustrate how we're using trying to combine cancer biology and cancer medicine with these increasingly powerful systematic big data approaches. We used it to discover that phosphate homeostasis, which hadn't occurred to me to be something to think much about in ovarian cancer, or any cancer for that matter, might be important, and that XPR1, the sole phosphate exporter, is potentially a new therapeutic strategy, specifically either targeting XPR1 or Kidneys 220 directly, or indirectly targeting XPR1 function through inhibition of inositol phosphate 6 kinase with a small molecule, that might be an effective therapeutic approach in the subset of ovarian cancers that highly express the phosphate importer SLC34A2. I also tried to show you how, how we're using this PRISM barcoding method to allow for highly efficient, cost-effective, high throughput profiling of both individual small molecules or potential future drugs of interest, to really understand what actually is their pattern of cell killing, not just across your favorite model or two or three, but across what we now know to be the enormous molecular diversity of cancer. So doing that over many hundreds of cancer models. And then I showed you this one anecdote of a class of compounds typified by topoxylin, which looks like it selectively kills tumors that are in this MDR1 drug-resistant state. And so we're exploring now whether there are optimized versions of topoxylin that could be brought to the clinic, particularly for treatment in those patients that have developed drug resistance, for example, to Taxol by virtue of MDR1 overexpression. And in preclinical studies that I didn't have a chance to show here, actually, if you select for models that acquire resistance to topoxylin, they do that by downregulating MDR1 and then resensitize to the thing they were originally resistant to, Taxol, for example. So you could imagine giving combination therapy either together or serially in a way that you can't escape both. You can escape taxanes if you're MDR1 low, high, sorry, but then you become sensitive to topoxylin or its analogs and vice versa. So I think that's something that we're trying to explore. Is there a feasible path to bring this therapeutic concept closer to the clinic? So I've tried to acknowledge the people that have done this work, in particular, Nick Wagley, who led the Count Me In team, which is continuing and growing at the Broad. Daniel Bondison has led the XPR1 ovarian cancer data, and Stephen Corsello and Channing Yu and Jennifer Roth and the whole PRISM team led the PRISM and MDR1 project. And so I thank you very much for your attention, and I'm happy to take questions. I will stop sharing my screen.
Fantastic, Todd. I will congratulate on everyone who's on the webinar Zoom. There's a lot of people. There's already quite a few comments in the chat, and I will take the first question myself. I have the ability to do that. And maybe the first question is a high-level question, and maybe it's a kind of some may say a little bit unfair of what's next. I think you'd pointed this out. One of the challenges with the cell lines is the growth on, 2D, and you look for intrinsic drivers of cancer. And I know you have a lot going on at the Broad, looking at next generation models or other models. Can you give us some insight on what you're excited about and what you think the next thing's going to be you guys are going to deliver for this type of analysis?
I don't know if we're going to deliver it, but I'll tell you what I think is important to think about. And that is, I think we tend to think of a trade-off between, okay, you can have your high throughput stuff on plastic, or, that's limited for all the reasons we know why it's limited, cell lines growing in plastic and high oxygen and high glucose and all that. Or you can do the deep mechanistic tumor microenvironment, engaging kind of studies more narrowly, but you can't have both. And so what I think we need to think about is, are there ways to get closer to being able to bring these kind of high throughput approaches, for example, all the way to the in vivo setting? So we're doing some experiments now to do in vivo CRISPR screens in the xenograft setting, with particularly asking the question, do there exist in vivo dependencies that are overlooked by the in vitro screens? And we already know the answer is yes. Maybe not a massive number of them, but they exist. And certainly we can also see that there are some in vitro dependencies that when you look in vivo, they go away entirely. And what aspect of the tumor microenvironment explains that, is an interesting question. Are all tumor microenvironments the same? Clearly not, because we see some cases where, the result that you get, for example, as a subcutaneous xenograft, which isn't exactly the most, most cancers don't metastasize subcutaneously. Some do, but most don't. Maybe very different than what the liver looks like, or the brain, or the lung, for example. So trying to bring these high throughput methods to the in vivo setting, in particular, looking at the components of the tumor microenvironment, I think that's sort of the next frontier.
There's a question, I was going to go in order, but there's a question down the list. I'll just ask this now. Are you doing that in immune competent models as well to look at IO effects?
So, yes, there's a project to do that in syngeneic transplantable models, where you have the benefit of immune competent mice, which is great. The challenge there is that there are like a half a dozen transplantable models. And so it's great, if you're using those classic models that everyone uses, but they don't model the diversity of human cancer that we know is just molecularly massive in its diversity. What we've started thinking about, and maybe people have experience with that on your end, is, is there something to be done with humanized mice that would allow you to bring the human models into an in vivo situation with at least some immunocompetence, even if it's not, exactly normal because it's a transplanted human immune system. Most of the work that we're doing in the sort of DepMap kind of context in vivo is in immunodeficient mice at the moment.
Yeah, there's investigators here doing the humanized models, but as you know, they're super expensive and it'd be hard to do screening in those. I can't imagine how you would do that, but maybe we'll get there.
Exactly.
So going back to the list, there's a question about data sharing. You mentioned this right at the start, actually, when I did my introduction about when you started, that wasn't the norm. And it still seems to be quite a challenge at the moment. If you think about how many metastatic breast cancer genomes are shared, it's difficult. What are the barriers you encountered, and how did you overcome them to have this ability to do this widespread data sharing?
Well, I think there are a couple of things. One is there was a fair amount of public shaming that we used just to say, look, it's just not right to not be sharing your data. And then we pressed on the journals to say, you should make it a requirement of publication. If someone's going to publish a big data set and then take some little slice of it and write a paper about it, and then save the rest of the data for another slice of a paper, you should say, no, we won't accept the paper unless you make it all publicly available. So I think that made progress. I actually feel like we've largely gone beyond the data hoarding instinct of the cancer research community. I mean, people talk about that a lot, but I think most people genuinely see the value in sharing data and are willing to do it. So what are some of the obstacles? Some of it is just technical. Not so easy to make data available in a usable format. The NIH has helped, but it's not so straightforward through the NIH. They, for example, have not agreed to pay for the hosting of genomic data for the community, for anyone who wants to make it available, whether it's like NCI funded or not. So I think we need to make it super easy for patients to say, I want to share, and we need to make it super easy for researchers to say, I want to share, by creating data platforms and a sustainable funding mechanism that supports data sharing. And I think increasingly, some of those technical solutions may also help companies feel more comfortable sharing data. I've heard from some companies who sit on a lot of useful data, related to clinical trials and so on, where they say, well, we have, this sensitive patient information, we're willing to share it, but we're worried about just like putting it out there and then having people downloading it and using it inappropriately. If we could have a secure solution that says, don't worry, it will be in secure hands. It will only be able, people can't just download it. They'll need to work with it on the cloud and in a way that doesn't, violate patient privacy and so on. I think that will help companies find their way to share more than they have.
Yeah, I do think one of the challenges, the regulatory burdens seem to have gone up tremendously. And I think that's because of a lack of understanding, this kind of fears of, misuse and all these things set off all these legal things, which then everything kind of grinds a lot of it to a halt, unfortunately. But I guess we'll move on for this. You gave a couple of examples in there of kind of accelerated drug ID to use in humans, one from the Count Me In, the angiosarcoma, and then also the one at the end. Is this one of your senses that one of your hopes that this sharing is going to accelerate the timeline to ID and then eventual use in humans, particularly, I guess, with the repurposing library?
I would say yes and no. I think these kind of approaches can be a powerful way to accelerate how much we know about a drug before we bring it into the clinic. And that can have a real benefit and maybe even result in, so we've heard of a number of drugs that were kind of headed towards the clinic, and then they profiled them in the DepMap, like in PRISM, and said, oh, that doesn't have anything close to the pattern of selectivity that we thought it did, this doesn't feel like it's ready to go forward yet. So that's one way that you have impact. Is that an accelerant or a decelerant? It depends on your perspective. I think with respect to like the repurposing, take a drug that's already been FDA approved and move it directly into a new clinical indication rapidly. That's a really attractive hypothesis. I think empirically, we've not had a lot of examples of that, maybe any yet, because most of the interesting new discoveries, new findings of old drugs have been either at concentrations that are higher than are the approved dose, which may still be tolerable, but if it's active at a dose that's higher than the approved dose, you can't just bring it back at that higher dose without doing further dose escalation. And even more commonly, most of the time that we see interesting activity of known drugs, it's explained by a mechanism that's totally unrelated to the mechanism that's on the bottle. So that's really interesting because, and that's where I think most of the power is of this repurposing library isn't necessarily repurposing per se, but it's using drug-like molecules or actual drugs to get to mechanisms that you might not get at with simple loss of function, like molecular glues or other types of functions that, loss of a scaffolding entirety of, just knockout might miss or knockout might obscure. So that's kind of what the current impressions.
Yeah. There's a question about the count me in that you showed. You showed this very interesting feature that many of the patients were one patient from like 1,200 different, hospitals. And the question is, how will that work in terms of tissue? That's okay for getting data, yeah, because it exists in the EHR and stuff. But presumably those single small county hospitals or whatever, or community hospitals don't have abilities to bank samples. So do you have ideas on how you're going to because it's the samples that's somewhat most key for precision medicine. Yeah, do you have an idea how you're going to do that?
So the tumor samples there are all FFPE blocks.
Yeah.
Or
so you would just hope technology comes good enough that in the end, FFPE might be the thing of choice, I guess.
Yes, and there will always be things you can't do with fixed tissues, but there's a lot that you can, and so that's all these things are a trade-off of scale versus technical feasibility. But I would also say you say clinical data, sharing clinical data is easy, but in the current state of things, sometimes that's like a doctor's office faxing a medical record and then somebody reading the medical record and abstracting it. That's going to change if only by, large language models and other, exciting things that are happening now. That problem of being able to access clinical data is going to go away. But at this exact moment in time, even that's not at all trivial.
Yeah.
And we need to fix that problem as well.
There was a question from Michael. I read this, Michael Lotz. I read it and I didn't understand it. It'd be interesting to see whether you understand this. What about applying a DNA encoded library against your pooled lines? Which I think he's saying, so you have these PRISM lines, and then I think saying doing a CRISPR screen in the PRISM lines.
Well, if it's a DNA encoded library of small molecules that are DNA, they're so
small molecules.
You could do that somehow in living cells, which I think my colleague Stuart Schreiber called DELs on cells, which we haven't really tried. Or you could somehow use, like protein extracts from the PRISM pool against a DEL library. At the moment, that's probably pushing the boundary of, what's technically possible, beyond what would be possible. Could you electroporate the Dell library into cells? Yeah, that's an interesting idea. I think I don't know that that's really been done routinely, even for a single cell line. One challenge is that on a pooling scale, it turns out to be an enormous number of cells. Like if you got 1,000 cell lines, yes, you can pool them all, but if you want to have representation of each of them, then in aggregate, it's still a large number of cells that can be, pretty unwieldy.
Yeah, Michael is always pushing the boundary. He likes to push the boundary, so that's good. Well, we are at 5 o'clock. Todd, I want to thank you for the speaker series, and I think everyone really enjoyed it. We had a large audience and lots of questions, and this was fantastic. And I'll be getting back with you.
Wonderful, great to be with you all.
Thanks for your time.
Bye.